Hi there, I'm Nicoletta, and I've successfully defended my PhD in computational toxicology. Because I was asked by several of you, including my family, friends, and colleagues, about the topic I've been studying for the past 3 years, I decided to come here and briefly explain it. So, take your cup of coffee or tea, take a break from your remote activities, and chill out for the next few minutes to find out more about my research work. Computational toxicology is the science that uses mathematical, statistical, and machine learning tools to study and predict potential toxic effects of chemicals. We are exposed daily to chemicals, for example, cosmetic products, pesticides in agriculture, or any industrial process. And we know only for a portion of them how safe they are. Be they novel or existing chemicals, there is a need to assess them appropriately and determine if any actions for protection are required. Just as we wish to keep the benefits of chemicals in our life, we must take care to minimize any harmful effects as well. Scientists, regulators, and other professionals agreed upon the need for a new, smart way to assess the potential harmful effects of chemicals. The call for a faster, cheaper, and more effective and reliable approach that does not involve animal testing, but rather a more human-relevant approach, is at the heart of a paradigm shift known as toxicology in the 21st century. However, the question remains, how to achieve this? My research project focused on neurodegenerative diseases, which are a series of diseases at the brain level characterized by the progressive degeneration of the structure and function of the brain. There is evidence that the brain is not completely developed until we are 25 years old, so the effects chemicals might have on the brain development are extremely important. If the structure or function of the brain is altered during development, this can have lifelong consequences for the individual and can also have societal impacts. Think of diseases such as Alzheimer's, Parkinson's, or learning disabilities such as autism and ADHD in children. So there is a need to understand and identify chemicals that induce damage to the brain to protect the developing brain from the exposure to harmful chemicals. The aim of my PhD was to formulate strategies using available information and computational approaches to develop models to screen chemicals for their ability to cause toxic effects to the nervous system. For that, I have to introduce to you a few additional terms. The adverse outcome pathway is a tool to organize the knowledge into a series of building blocks that describes an adverse effect that occurs at various levels of biological organization. Imagine a chemical that binds to a protein or receptor at the molecular level. Because of this binding, the chemical can induce an effect at other biological levels, such as cellular, tissue, organ, organism, and population levels. It is similar to the domino effect, a chain reaction induced by chemicals or other stressors such as radiation, nanoparticles, or pathogens such as viruses. And most importantly, the AOP is a way to explain causally how a chemical induces a harmful effect. Networks are graphical models used by scientists to represent the connections and relationships between all possible linear biological paths. Such models help to explain how proteins and effects interact with one another. We all live in a world full of networks, and so biology represents a huge network as well that needs to be understood. It can get really complicated. What did I do as part of my PhD project? I collected linear adverse outcome pathways that describe toxic effects at the brain level from the knowledge base, a database maintained by the Organization for Economic Cooperation and Development, OECD. I then developed a network of adverse outcome pathways and analyzed it mathematically. This analysis allowed to identify most connected biological events in the network. 3 of such biological events included reduction of the protein named brain-derived neurotrophic factor, BDNF, decrease in the formation of synapses, defined as synaptogenesis, and decrease in the function of neural network. I then collected and curated data obtained in the labs and data generated by computers. This data collection allowed for the development of a computational model to quantify the simplified causal biological path formed of the three most connected biological events. Furthermore, the model was able to predict the probability that a chemical induces each biological event and ultimately alters our brain. This kind of modeling is more informative than classical statistical approaches. It describes the uncertainty given by the data and mathematical assumptions, leading to a transparent, traceable, reproducible, and reliable assessment. Luckily, efforts to investigate and simulate our key events of the network are ongoing. Hopefully, in the near future, we will know more about neurotoxicity, and such novel practices will be increasingly applied and accepted by regulators. This will add value to the chemical safety assessment, contributing to the paradigm shift in the 21st century toxicology, and ultimately safeguard the human health. These results wouldn't have been possible without the support from my supervisors and collaborators. Thank you for watching.